Hello, I'm Essay, your AI essayist. Please note that all content, audio and video in this presentation are AI generated. As AI can sometimes produce inaccuracies, I encourage you to verify any factual information presented. With that in mind, let's begin.
The 1984 Nobel Prize in Physiology or Medicine recognized a monumental achievement, the creation of monoclonal antibodies. César Milstein, Köhler, and Niels Jerne unlocked a technology that fundamentally altered biomedical research and clinical practice. Prior to this innovation, antibodies, the immune system's targeted weapons, existed primarily in heterogeneous mixtures known as polyclonal antibodies. These mixtures, derived from multiple B lymphocyte clones each producing a distinct antibody, presented challenges for research, diagnostics, and therapeutics. Their inherent variability, limited specificity, and batch-to-batch inconsistency hindered precise experimentation and clinical application. The monoclonal antibody offered a definitive solution, an unlimited source of identical antibodies, each precisely targeting a single antigenic epitope. Building on the shoulders of giants, Milstein and Köhler's work cannot be fully appreciated apart from Jerne's theoretical contributions. Jerne's network theory of the immune system, a radical concept at the time, proposed that the immune system exists as a self-regulating network of interacting antibodies and lymphocytes. This theory, while initially met with skepticism due to the limited experimental tools available for its complete validation, suggested an intricate level of antibody diversity and regulation previously unappreciated. Jerne posited that antibodies did not merely react to foreign antigens, but also interacted with each other, forming a complex network of stimulatory and inhibitory signals. This framework inspired the quest for methods to isolate and characterize individual antibody specificities, a goal that monoclonal antibodies would ultimately, for Jerne's original conception, though groundbreaking, lacked the experimental verification that would solidify its place in immunological dogma. His theory predicted a complex web of interactions, yet the technology to dissect and analyze these interactions at the molecular level remained elusive. Monoclonal antibodies provided the key to unlock these complexities, offering the ability to probe the immune network with unprecedented precision. Researchers could now generate and study individual antibodies, testing Jerne's theoretical predictions and refining the network theory based on empirical evidence. The initial theory contained a set of differential equations describing the rates of change of antibody populations and their influence on each other. Experimental validation of these equations was previously impossible. The central problem elegantly addressed by monoclonal antibodies was the production of unlimited quantities of highly specific antibodies. Polyclonal antibodies, harvested from immunized animals, represent a transient response, varying in composition and potency over time. This variability complicated research efforts requiring reproducible results, as well as clinical applications demanding standardized reagents. Milstein and Köhler's methodology provided the key. A technique involved fusing myeloma cells, cancerous plasma cells possessing the property of immortality, with antibody-producing B lymphocytes derived from immunized mice. This fusion created hybridomas, immortalized cell lines capable of producing and secreting monoclonal antibodies indefinitely. The process involved several critical steps. First, mice were immunized with a specific antigen to stimulate an immune response. Splenocytes, containing antibody-producing B lymphocytes, were then harvested from the immunized mice and fused with myeloma cells using polyethylene glycol or other fusion agents. The resulting mixture contained a heterogeneous population of cells, including unfused B lymphocytes, unfused myeloma cells, and hybridomas. A selection process, still involving the use of a selective medium like HAT hyperxanthine, a monopterol of thymidine, eliminated the unfused myeloma cells and B lymphocytes, leaving only the hybridomas to proliferate. The subsequent crucial step involved screening hybridomas to identify those producing antibodies of the desired specificity. This was typically achieved using enzyme-linked immunosorbent assay, where the target antigen was immobilized on a solid support and the antibodies secreted by the hybridomas were allowed to bind. Hybridomas producing antibodies that bound to the antigen were then selected for cloning and further characterization. The screening process, while conceptually simple, required significant effort to optimize and perform at scale. An early and impactful application of monoclonal antibodies arose in blood typing. Traditional blood typing relied on polyclonal antisera, which occasionally exhibited cross-reactivity and batch variability. Monoclonal antibodies, with their exquisite specificity, improved the accuracy and reliability of blood typing procedures, minimizing the risk of transfusion reactions. Furthermore, Monoclonal antibody provided a powerful tool for identifying and characterizing cell surface markers, enabling the classification of lymphocytes and other immune cells into distinct subsets. This, in turn, facilitated a deeper understanding of immune cell function and disease pathogenesis. The immediate impact of monoclonal antibody technology resonated throughout basic research. The ability to generate antibodies against virtually any target molecule empowered researchers to identify and characterize previously unknown proteins and cellular processes. Monoclonal antibodies became indispensable reagents in techniques such as immunofluorescence microscopy, allowing researchers to visualize the distribution of specific proteins within cells and tissues. The intellectual climate of immunology at the time was charged with excitement, with laboratories worldwide rapidly adopting monoclonal antibody technology and exploring its vast potential. The initial reception was, however, marked by a degree of skepticism in some quarters, concerns regarding the robustness of the hybridoma technology, and the potential for genetic instability in the immortalized cell lines were raised. These concerns diminished as the method was validated through widespread adoption and successful application. Milstein and Köhler asserted that unlimited quantities of identical antibodies can be produced using myeloma lymphocyte fusion to create hybridomas that can be cultured indefinitely and secrete monoclonal antibodies. This claim, initially viewed with caution, found subsequent confirmation with their technique's widespread adoption. The monoclonal antibody technology, born from a synthesis of Jerne's theoretical immunology and Milstein and Köhler's innovative cell fusion techniques, revolutionized biomedical research and laid the foundation for a new era of targeted therapeutics. The advent of monoclonal antibodies ushered in an era of unprecedented precision in biomedical research, particularly in diagnostics. Enzyme-linked immunosorbent assays, commonly known as ELISA, experienced a paradigm shift. Prior to monoclonal antibodies, ELISA relied on polyclonal antibodies, which suffered from batch-to-batch variability and a lack of specificity. The introduction of monoclonal antibodies provided a homogeneous, highly specific reagent, enabling the development of more reliable and quantitative ELISA. This advancement was crucial for the accurate detection and quantification of a wide range of analytes, from hormones and cytokines to infectious disease markers. Western blotting, another cornerstone technique in molecular biology, also benefited significantly from the advent of monoclonal antibodies. The ability to generate antibodies that bind to specific epitopes within a protein enabled researchers to identify and characterize proteins with greater accuracy. Moreover, the use of monoclonal antibodies in immunohistochemistry revolutionized the field of pathology by targeting specific proteins expressed in tissue samples. Pathologists gained a powerful tool for diagnosing and classifying diseases, particularly cancer. For instance, the identification of specific tumor markers using monoclonal antibodies facilitated the development of targeted therapies and improved patient outcomes. Monoclonal antibodies also spurred advancements in therapeutics, initially directed towards novel antibody-based cancer therapies. The initial steps to harness monoclonal antibodies centered on the murine form of such antibodies. The murine form created challenges associated with their immunogenicity in humans. Early clinical trials revealed that patients treated with murine monoclonal antibodies often developed human anti-mouse antibody responses, limiting their therapeutic efficacy and causing adverse effects. This obstacle forced the development of humanized and fully human antibodies. The process of humanization involves grafting the antigen-binding regions, also known as complementary titamating regions, from a murine monoclonal antibody onto a human antibody framework. This strategy significantly reduces the immunogenicity of the antibody while retaining its target specificity. Free human antibodies, generated using transgenic mice or phage display techniques, represent the pinnacle of antibody engineering. These antibodies are indistinguishable from naturally occurring human antibodies, minimizing the risk of immune responses and maximizing therapeutic efficacy. Characterizing antibody binding affinity and specificity requires employing mathematical and statistical methods. Techniques such as surface plasmon resonance and bilayer interferometry provide quantitative measurements of the association and dissociation rates of antibody-antigen complexes. These measurements can be used to determine the equilibrium dissociation constant, which is a measure of the affinity of the antibody for its target. A lower equilibrium dissociation constant indicates a higher affinity. The analysis of hybridoma cell lines also relies on mathematical and statistical tools. Limiting dilution assays are used to isolate single-cell clones that produce the desired monoclonal antibody. The statistical distribution of antibody secretion levels across different clones can be analyzed to identify high-producing cell lines. Mathematical models can be used to predict the long-term stability of hybridoma cell lines and optimize culture conditions for antibody production. Beyond diagnostics and therapeutics, monoclonal antibodies played a pivotal role in advancing our understanding of immune system regulation and disease pathogenesis. Jerne's network theory, which posits that the immune system is a self-regulating network of interacting antibodies, laid a theoretical foundation for the development of monoclonal antibody technologies. The network theory proposes that antibodies can recognize and bind to other antibodies, forming a complex web of interactions that maintain immune homeostases. Within the network theory, mathematical formulations describe the dynamics of antibody interactions. For example, differential equations can model the production, clearance, and binding of antibodies within the network. These mathematical models can be used to predict the effects of perturbations, such as vaccination or infection, on the immune system. Mathematical modeling has also been instrumental in understanding the phenomenon of idiotypic regulation, where antibodies recognize and regulate the expression of other antibodies with similar antigen specificity. Monoclonal antibodies enabled researchers to dissect the intricate mechanisms of immune regulation and identify key players in disease pathogenesis. By targeting specific immune cells or signaling molecules, scientists gained new insights into the causes of autoimmune diseases, infectious diseases, and cancer. This knowledge has paved the way for the development of novel immunotherapies that harness the power of the immune system to fight. The monoclonal antibody technology is a cornerstone of modern biomedicine. Its impact resonates throughout diagnostics, therapeutics, and basic research. The ability to generate unlimited quantities of highly specific antibodies revolutionized our ability to diagnose and treat diseases, and to understand the inner workings of the immune system. The enduring legacy of Milstein and Köhler's work lies not only in the technological innovation itself, but in the paradigm shift it engendered, emphasizing the power of combining theoretical insights with innovative experimental techniques to revolutionize scientific field. The original monoclonal antibody technology underwent refinement. Efforts continue to improve antibody engineering and therapeutic efficacy. These include the development of bispecific antibodies, which can simultaneously bind to different targets, and antibody-drug conjugates, which deliver cytotoxic payloads directly to cancer cells. The transformative impact of monoclonal antibodies extends beyond their immediate applications, shaping the trajectory of biomedical research and paving the way for a future where targeted therapies are tailored to the individual patient.
We really appreciate you tuning into this AI-powered presentation. Our goal is to push the boundaries of AI in content creation, and your input is invaluable. If there's a particular subject you'd like to see us tackle next, or if you have suggestions on how we could fine-tune our AI's creative prompts, please don't hesitate to leave your ideas in the comments section. We're excited to bring you more unique content very soon.